Hello, my name is Carla Garcia, and here's my presentation on the five senses. So all of our senses have to go through sensory transduction, which is converting various forms of energy outside of the body into neural signals in order for our brain to understand them. So here are all the different types of energy. So for sight, it's photons, which are light rays. For smell, it's different odors. For hearing, it's pressure waves in the air, which is sound. For touch, it's either pressure, itch, or vibration on our skin. And for taste, it's different food particles that have these different tastes. So the main thing about how our senses work is all in the neurons, which are specialized cells designed to transmit information. So now I'm going to explain how we're able to use each of the senses. So first we have sight. So rhodopsin molecules, which are gene protein coupled receptors, located in the outer segments of photoreceptors, undergoes conformational change when struck by a photon. So these are the photoreceptors. They're located in the retina of the eye. These are rods and these are cones. So then the activation of the rhodopsin then activates the gene protein transduction, which is a process that converts light into electrical signals. And then the phototransduction cascade is initiated, which amplifies the initial signal of the photon of light. So next we have smell. So a molecule in an object such as a rose enters the nasal cavity and then activates a bunch of different receptors. When the receptors are activated, the olfactory sensory neurons are activated, and those neurons then activate a region in the olfactory bulb, which is right here, uh, which is responsible for detecting certain chemicals. For like in my example with the rose, it can smell the rose and identify that it's the smell of a rose. Okay, next we have taste. So taste buds on our tongues have receptors that can be stimulated by food molecules. So this would be a molecule and this would be a taste bud. So taste receptors are G protein coupled receptors, just like the rhodopsin molecules, which is used in sight. So the taste receptors activate TRP channels that produce a short depolarization. And then the impulse of this from the neuron is sent to the gustatory cortex in the brain. And you can see the taste receptor goes into the nerve and goes to the brain. So just like smell, there's a chemical stimuli that activates both the olfactory, which is from smell, and the gustatory system. Now we have hearing. So sound waves enter our ear and they pass the eardrum and it reaches the cochlea, which is this spiral thing, and pushes on hair cells. Mechanoreceptors on the hair cells trigger action potentials based on the direction of the movement of the cells, and this is a mechanical stimulation. The hair cells in the cochlea are organized as a tectonic map, so the different levels of stiffness make it differently sensitive to different frequencies of sound, and so the brain is able to recognize the frequencies. So the activated part of the cochlea then sends action potentials to the primary auditory cortex in the brain. And then the last sense is touch. So there are touch receptors, which are sensory neurons, that are located in the epidermis and dermis of the skin. So the epidermis is the top part of the skin, and the dermis is the underlayer. And all the different receptors are scattered throughout the skin. So the receptors have different size receptive fields. So if they have smaller fields, it means it's more sensitive to touch or say heat or pain or pressure. So they are responding to a mechanical stimulation, which hearing also handles. The information is sent to the brain through different sensory fibers. And then the homunculus is a topographic organization of how certain parts of our body is represented in the brain. More sensitive areas, such as our hands, have a bigger representation in the brain. And thank you, that's all.